Hello everyone, welcome to the new session of Quantitative Inheritance Part 2. So in the first part, we have discussed about the quantitative inheritance with some examples by taking example as kernel color in case of wheat plant, right? In case of wheat plant. So we will take one more example to understand this quantitative inheritance in better way. Skin color in man. We are taking here example of skin color in man. So a polygenic inheritance. Polygenic inheritance means many genes are involved here. So multiple gene inheritance is also called as a polygenic inheritance and was given by the Davenport 1913 in Jamaica. Okay. So he found that two pairs of gene, that is capital A, small a, capital B, small b, cause the difference in the skin pigmentation. Okay, so these genotypes were present in the Negroes and Caucasian people. So in the Negroes, capital A, capital B, whereas in the case of Caucasian people, they have small a, and small b type of genotype. Okay. So these genes were found to affect the character in the additive fashion. Which genes? The genes which is present in the Negroes and Caucasian people. That is capital A, capital B in the case of Negroes, and small a, small b type in white or Caucasian people, right? So these genes were found to affect character in additive fashion, right? So thus true Negroes has a dominant gene. So this one is the dominant gene. Negroes is having the dominant gene, right? And the white Caucasian is having the recessive gene. So this is the recessive gene. Okay. So the F1 offspring of the mating, that is small a, small b, small b with capital A with capital B are all capital A, capital B, small b type. How it happens? Let us see. So the Negroes is having genotype capital A, capital B, okay, and the Caucasian will have small a, small b type. This is the Negroes. This is the Caucasian one, right? So they produce single type of gametes, that is capital A, capital B, okay, and they will produce, Caucasian will produce a gamete that is small a, small b type. Now when there is a fertilization, then they will produce capital A, capital B, small b type. Okay, so they will produce, all will be capital A, capital B, small b type. Okay, all the offspring will be this type and have an intermediate skin color termed as mulatto. Now this is dark skin, and this is white skin. Now this is having an intermediate between the dark skin and white skin, and that is called as mulatto. Okay, so a mating of the two such mulattoes. Now in the second generation, okay, for the F2 cross, we will take mulattoes as an example. Okay, so a mating of two such mulattoes produces a wide variety of skin color in the offspring, ranging from the skin as dark as the original Negro's parent and as white as the original white parent. So there will be original Negro's parent like offspring and also offspring with original white parent skin. Okay, so let us understand with the taking this as a cross. So Negroes is having capital A, capital B type genotype, whereas the white or the Caucasian will have small a, small b type. Now they will produce gamete as capital A, capital B, and here they will produce gamete as small a, small b. So these are the two gametes in the case of Negroes and white. Okay. Now they will produce mulatto. That is the intermediate skin color between the Negroes and white. So it will be like this. This will be mulatto, right? Now we will take mulatto, one mulatto as a female and one mulatto as a male, right? And we will see what happened in the next generation cross. So they will produce different types of gametes. So if we want to see the gametes, how they will produce, capital A, right? So there will be gamete like capital AB, capital A, small b, small a, capital B, and small a, small b. Okay, so there will be 4 different types of gametes produced in the F2 generation. Okay, so this will be the phenotypes. So we will have black, that is the parental one, and the white, that is also showing the parental one. Now we will have an intermediate, that is the mulatto. Okay, and we will also have a dark and light shade. So this is the type of genotypes we will have in the cross. Okay, that is the parental one, and here we will have the intermediate form, but these are 2 other skin color, that is the dark and light type also produced. Okay, so genotype frequencies also here we can see. The parental one is just 1 and 1, both the type, and we will have 4 and 4, that is the dark and light, and 6 individual be of intermediate type. Okay, so these results are clearly showing that A and B genes produce about the same amount of darkening of the skin. Okay, so how much the A is producing the pigment, the same B will also be produced that amount of the pigment. Okay, so same amount of darkening of the skin. Therefore, increase or decrease of A and B genes causes the variable phenotypes in the F2 in the ratio of 1 Negro, 4 dark, 6 intermediate, and 4 light, and 1 white. So how and why it happened, now you can understand, because we can have understanding like this, and here we can have understanding like this, right? For the dark we can have like this. Okay, so this is having this dark. Now in the other case they can have like this, right? Or they can have like this. So depending upon the dose of genes, 2A or single A in the form of capital 1 and small 1 also, capital B or small b also. So what type of doses of the genes they are having, depending on that they will be Negroes, dark, intermediate, light, and white type of skin formation. Okay, so there is a equal contribution of A and B genes. Okay, so if both the A is capital form or one is small cap B and small, there will be a capital B, then you can understand then this will give a more dose of dark skin as compared to this one, right? Skin color in man is a rather simple example of polygenic inheritance because only 2 pairs of genes are involved, right? We are having only 2 pairs of genes. So the inheritance of height in man is more complex phenomena involving perhaps 10 or more pairs of genes. So this one is more complex. This is also an example of the polygenic inheritance, the height in man. But here it is more complex because 10 or more pairs of genes are involved, right? So the character of tallness is a recessive to shortness. Thus, an individual having the genotype of more dominant gene will have the phenotype of shortness. So if the gene is more dominant, then the height will be of short type. And if the gene is of more of recessive type, then the man will be tall. That's showing the tallness character. Okay, so this quantitative trait is controlled by multiple pairs of gene, and it's fairly influenced by the variety of the environmental factors also. If one measured the height of 1,000 adult men and height of each is plotted against the height in centimeters and the point is connected, a bell-shaped curve is produced. So 1,000 peoples are taken as a sample, right? And we measured the height and we plot. Then we can have such type of plot. So this curve is called as curve of normal distribution, and this is the characteristic of the quantitative inheritance. So this is the normal distribution, curve of normal distribution. So if one measures the same way, if one measures the length of 1,000 seashells, the same species, or counts the number of kernels per year in 1,000 ears of corn, right? So, ear, this is the kernel, right? This is called ear, kernel color in wheat. So this is kernel color. That is the ear of the corn and the wheat also. If we see the corn, it will be like this, right? It will be like this. So kernel color per ear in 1,000 ear of corn, or the kernel color of wheat, or the numbers of pigs per litter in 1,000 liters, okay, or the weight 1,000 hen egg, one will find a normal curve distribution. In every case, in all the given example, we will have a normal bell-shaped curve like this. 1,000 in a sample of 1,000, whether you're taking an example of the seashell, whether the kernels per ear of wheat or the corn, okay. Pigs, taking examples of pigs, okay, 1,000 hen's egg, you will have in all the case this bell-shaped curve. That is the normal curve of distribution. The easiest character or the traits to deal with are those involving the discontinuous or a qualitative differences that are governed by one or a few major genes. So discontinuous, that is the qualitative inheritance. We have seen in the case of qualitative inheritance that involve discontinuous variation, right? So many such inherited differences exist, and they frequently have profound effect on the plant's value and their utilization. So such type of differences or such type of pattern of inheritance that can be seen in the plant value and their utilization. How frequently they have profound effects on plant value and their utilization. Examples are the starchy versus sugary kernels. That is the characteristic of the field of sweet corn respectively. So sweet corn are can either be a starchy one, or that can be having a sugar, that having the more sugar content. And determinants versus indeterminate habit growth in the green beans. Okay, that is the determinant varieties or adapted to the mechanical harvesting. Such differences can be seen easily and evaluated quickly, and the expression of the traits remains same regardless of the environment in which the plant grows. So traits of this type are termed are highly heritable. So these are highly heritable because there is expression of the trait remain the same regardless of the environment, no effect on of environment. So they are highly heritable. A qualitative trait is expressed qualitatively, which means that a phenotype falls into the different categories. So we are talking about the qualitative traits. So they are expressed qualitatively, right? Which means that phenotype falls into a different categories. There is a clear-cut demarcation. This we have already discussed in our previous session. So these categories do not necessarily have a certain order or certain spectral type of things that we have seen in the previous session. The pattern of inheritance for the qualitative trait is typically monogenetic, which means that the trait is only influenced by a single gene. Single gene is responsible. So inherited disease caused by the single mutation or a good example of the qualitative trait. Okay, so let's have a difference between the qualitative genetics and the quantitative genetics. This we have already discussed individually. Now see in a tabular form how the qualitative genetics differ from the quantitative one, right? So it deals with the inheritance of traits of the kind that is form, structure and color. So we have seen the form of seed, that is the round shape or the wrinkle shape, for example, that is the form, structure and color. Now, the quantitative genetics, it deals with the inheritance of traits of the degree, that is the height and the length, weight and number, also the skin color, right? So in this case, that is the qualitative genetics, it follows the phenotype shows the discontinuous variation, whereas in the case of quantitative genetics, a spectrum of phenotypic classes occur with contain the continuous variation, right? Each qualitative trait is governed by two or many alleles of a single gene. So here single gene is involved. Single gene is involved. And in this case, multiple gene is involved. Multiple genes are involved. Okay, so each quantitative trait is governed by many non-allelic genes or the polygenes. The phenotypic expression of a gene is not influenced by the environment. So there is no influence of the environmental factor on the phenotypic expression. Whereas in the case of quantitative one, okay, the environmental condition affect the phenotypic expression of the polygenes variously. So qualitative, it concerned with the individual mating and their progeny. It concerned with the population of organisms. So here we can see a curve for the population. So in this case, no such curve is obtained. And it is the analysis made by the count and ratios, but here we will have some statistical method for the analysis. So this is all about this session. We discussed about the quantitative inheritance. Okay, so I'm closing this session. Thank you very much.